milk, spice, and everything nice. But Professor Uranium accidentally added an extra ingredient to the concoction, coffee bean X. Thus, the ultimate coffee bean was born, using its ultra super taste. Okay, that's quite enough of that. Hi, you can call me Rainy, and today we're going to talk about the one and only C. arabica. So what's with this plant that makes it so special? Well, the answer is in its genes. The C. arabica is a tetraploid species. This means that a C. arabica cell carries 4 copies of chromosome in contrast to its parents, which are dark plots. C. arabica is also an alloploid, which is when its copies of chromosomes originate from different species. Combining all of this information, C. arabica is ultimately called an allotetraploid. The key takeaway from this is that C. arabica is a result of a polyploidization event, stably inheriting subgenomes from both C. canephora and C. Eugenioides. However, scientists found out that C. arabica has very low divergence between its species due to a severe bottleneck after the polyploidization event occurred all the way back in East Africa. So, is this good news or bad news? Well, it's bad. Really, really bad. Why? With the low genetic diversity of C. arabica among its population, the species becomes very susceptible to plant pests and diseases, especially to the coffee leaf rust disease. This creates a need to identify and investigate the C. arabica cultivation from its white diploid relatives to create a more resistant breed while maintaining a good beverage quality. Hence, only just a few decades ago, a Timor hybrid C. arabica breed was found with a C. Canephora introgression gene region as a product of a spontaneous crossover at the island of Timor. This introgression region was found on chromosome 4. The region is a set of combination of 3 different gene clusters, which are R genes that produce R proteins that provide the coffee leaf rust resistance, negative regulator of R genes that prevents translation of R genes to specific conditions, and leaf rust 10 disease-resistant locus receptor-like protein kinase, which acts as the positive regulator for the resistance response. This protein is also first discovered in wheat plants to resist striped rust fungus and powdery mildew. However, this introgression gene region reduced the beverage quality of the Timor hybrid line. Through homologous exchange of different subgenomes in C. arabica, scientists found out that there is an allelic bias in the sub-CC genome. There is an outlier observed in Caribbean typical cultivar where the bias is on sub-EE genome instead, due to a major crossover event. This feature helps in contributing to the phenotypic variability in C. arabica's small population genome, even though there is not much in the overall genetic diversity. Using the homologous exchange information, scientists have discovered that the first polyploidization event occurred as long as 610,000 years ago. Ever since then, multiple bottlenecks have occurred, creating 3 main genetic groups of C. arabica, namely the typical Bourbon cultivars, wild types, and Timor hybrid-derived cultivars. Hence, modern cultivars may be of a sole group or an admixture of them. Here is a highly simplified pedigree chart illustrating the type of relationships between the main groups of C. arabica. And for readability's sake, not all members are shown. The colored bubbles represent a cluster of C. arabica wild types and cultivars, and not all cultivars are bubbled. The line shows the type of relationship between them, with the straight line being parent offspring, the dashed line being grandparent offspring, and the dotted line to be great-grandparent offspring. Feel free to pause the video to take a closer look at it. For the final act, let's meet the Gesha cultivar, which originates from the forests of the Gesha region from the western Ethiopia. Being the current champion of the C. arabica cultivars, it has such a unique and enticing aroma compared to the others, but also being highly expensive. This is because this cultivar requires extremely specific cultivation conditions, such as cooler and higher altitudes and nutrient-rich volcanic soils. So, which new cultivar will be next to claim the title as the most perfect C. arabica cultivar in the world?